Okay, so in this section, we're going to take a look at some of the other levels of protein structure. So as a reminder, in the previous section, we looked at the primary structure, primary protein structure, and what that is is just the sequence of the amino acids. And when you have an amino acid in a protein, sometimes you'll see the term residue being used to reference those. Right? Residue is the same thing as an amino acid that you would find in a protein. So the primary structure is just listing those amino acid residues from the N terminus to the C terminus. So now we're going to take a look at the secondary structure. So secondary, as we saw before, is abbreviated to with the degree sign. So the secondary protein structure is actually determined by the hydrogen bonds between the peptide backbone of the protein. So for example, I have a pretty simple protein drawn up here, or a polypeptide. So on one end we have our N terminus and we have our C terminus on the other side, and we have our R groups in green here that would tell us which amino acids we have, and those R groups are all attached to the alpha carbon. So what we've highlighted in yellow would be the peptide backbone, right? So if you look all the way across, this backbone of the peptide bonds, right? So we have a peptide bond here, we have another peptide bond here, and if we continue, we would have another peptide bond over on this side. So what can happen, right? Remember, this oxygen here has a slight negative charge, and this H here can have a slight positive charge because it's attached to a nitrogen. So what can eventually happen then is this backbone can curl up or fold up as a result of hydrogen bonding between the peptide backbone. All right, so what you'll see here is that we have the conditions necessary for hydrogen bonding to exist. We have an oxygen here that is slightly negative, and then we have our hydrogen over here that is going to be slightly positive. Obviously, they can't hydrogen bond with each other, but if we had a long peptide or protein chain, amino acid residues further down the chain could actually hydrogen bond. And that is illustrated by our first secondary structure, which is known as an alpha helix. And you can think of an alpha helix as simply similar to a spiral staircase, but obviously not as big, right? This is on the molecular level. So if you notice here, there's our spiral staircase going all the way up. And why is it in this form to begin with? Well, it's held in place because of those hydrogen bonds, which we'll highlight here in green, that are going to occur now between the backbone peptide bonds of this particular protein, right? So you'll see your hydrogen bond here, for example, right? And that keeps that spring-like structure intact, right? So spiral staircase or a spring would be the analogous shapes that we can use to illustrate this alpha helix, right? Alpha helix is often shown as this. All right, so that's the first type of secondary structure, and that results from hydrogen bonding among or between the peptide backbone. The second type of secondary structure we'll see is something known as a beta pleated sheet, and this also occurs because of hydrogen bonding. Occurs due to It also occurs due to hydrogen bonding between the backbone peptide bonds, but in this case, what actually happens is your protein, if your protein starts down here, and the N-terminal end of our protein, right, it can continue down this way, and then it can fold back upon itself, and then folds back upon itself again. So what would then keep this shape or this structure intact? Well, that's due to the hydrogen bonding that can occur between, again, the peptide backbone, which let's highlight that in green. There's your peptide backbone, snaking all the way around and coming back around this way, and then snaking all the way around again. All right, so here you can see that we have a C double bond. Oh, there's your carbonyl, and there's a hydrogen attached to a nitrogen, and then this here would be a hydrogen bond, right? And that keeps this sort of snake-like or this serpentine structure intact. And the specific shape that it adopts is sort of like a sheet that's sort of puckered, or it looks like a zigzag shape, right? So that's known as a beta pleated sheet, and then the symbol for beta looks like that. So a beta pleated sheet, or just a beta sheet, would be the second type of secondary protein structure that we're going to see. And again, this happens because of the same type of hydrogen bonding that occurs between the backbone peptide bonds. And I'm highlighting the fact that it's hydrogen bonding between the backbone peptides because we're going to see hydrogen bonding later on that contributes to a different type of protein structure. Another type of secondary structure that you'll see is known as a triple helix. All right, and a triple helix, as the name suggests, would have 3 helices wound together. Right, so you can think of it sort of like how the strands in a rope are wound together to make a really, really tough rope in the first place. So here we have 3 of these sort of strands or polypeptide chains being woven together, and again you'll have hydrogen bonding holding those chains together in the first place. So that type of structure then would give a lot of strength, and you'd find these types of proteins in places like collagen and your skin and connective tissue, places where strength is really needed. So just to backtrack a second, when you look at I think some of your questions on the homework may have pictures like these. So often you'll see your alpha helix being represented sort of like a spiral or a spring like this, and then your beta pleated sheet would sort of look like just a bunch of arrows this way, right? So depending on whether you look online or elsewhere, you'll see this beta pleated sheet being represented this way with this sort of serpentine type structure, whereas the alpha helix is usually represented with a spring-like structure or a spiral staircase. All right, so the third level of protein structure is called tertiary. That's how we abbreviate tertiary. And what the tertiary structure is, we can think of it as the 3, so this is the relationship. Tertiary, third level, represents the 3-dimensional or the 3D shape of a protein. Right? How does it actually look in 3 dimensions? And that would be caused by all sorts of different types of interactions between different sections of the protein chain itself that would actually cause that protein chain to bend and twist and make a particular shape. Now the key thing that is going to, or things that are going to dictate this 3-dimensional or tertiary structure are known as cross-links, and also we're going to see some attractions and repulsions, and these here now are going to happen between the side chains or the R groups of the amino acids. All right, so we can say then that the 3D structure, the 3-dimensional structure, is determined by R group interactions. Okay, we talked about the R groups back when we looked at the amino acids in the first place. Now we're going to see how these R groups are able to interact with each other, which will then lead to the protein folding into a particular three-dimensional shape. All right, so there are several types of interactions that we're going to see that can dictate the three-dimensional shape of a protein. Before we get to that, I just wanted to show you really quickly. I tried to draw a diagram earlier on to depict an alpha helix versus a beta pleated sheet. This is a much better depiction of that. So here in red, these little spring-like sections, these are your alpha helices. Right, you can see they look like helix or helices, and then the blue flat regions, right, that are typically next to each other, these are your beta pleated sheets. Right, so you'll see that quite commonly in your textbook and elsewhere. So let's take a look now and see how the tertiary structure is determined and the types of interactions that we're going to see that drive the three-dimensional structure. So these R group interactions, we're going to see 5 of them. 5 types of R group interactions. And we're going to focus on those R groups. So the first one is known as a hydrophobic interaction. And the term hydrophobic here means water hating. And what that would mean by extension would mean that these R groups would have non-polar R groups. So these amino acids would have non-polar, right? Non-polar means they're not polar, which means that they're the opposite of polar. So if something is non-polar, it means it's not going to like water. So these hydrophobic interactions are going to occur between non-polar amino acids. You have the opposite of that, known as hydrophilic interactions, right? Hydrophilic here would mean water-loving. Right, and these would happen between the external aqueous environment, whether it's in the cell or outside of the cell, on the R groups now of polar amino acids. We're also going to encounter things known as salt bridges, and a salt bridges is just a fancy word for an ionic bond, and this is going to occur between R groups that are going to have positive charge and negatively charged R groups. And as we saw before, the positively charged R groups are going to be your basic, and your negatively charged R groups are going to be your acidic amino acids. So basic would be positive, and then your acidic ones will be negatively charged. So the coming together of those positives and negatively charged R groups is what a salt bridge is called. We're also going to see our good friend hydrogen bonding, right? That is going to occur between the hydrogen of a polar R group and the oxygen or the nitrogen of another amino acid. And then the third one, the last one here is the type of a bond, a covalent bond known as a disulfide bond, which we saw back in, seems like a few chapters ago. And if you remember, a disulfide bond happens when you have 2 thiols like that can then interact with each other, and you'll end up losing hydrogens, and you end up then making that disulfide bond between them. So the only amino acid that has an SH as a part of the R group are your cysteine amino acids, right? These are going to contain that SH, that thiol group that can participate in a disulfide bond with another cysteine residue. So if we go back to our molecule that we showed above, we can now identify some of these R group interactions. All right, so the first one we can take a look at here is your disulfide bonds, right? And it's pretty easy to pick that out. It's going to have two sulfurs linked together, and those are going to happen between, again, cysteine amino acid residues. Here on the left side, we have a hydrogen bond occurring between one polar amino acid, the hydrogen of one and the oxygen of another one. We have a salt bridge, which is simply a positive charged R group being close to a negatively charged R group. We also have our hydrophobic interactions, which are over here. We have our non-polar amino acids with their R groups associated with them. And then the fifth one that we saw would be the hydrophilic interaction with water, right? So here we have our polar amino acid R groups that will interact with the aqueous environment on the outside. All right, so these are going to be the 5 types of R group interactions that are going to drive the 3-dimensional shape of our protein. Okay, so if you were given, for instance, a couple different amino acids and you had to determine the type of tertiary or R group interaction, right, you would have to look up their structures from your table, from your amino acid table, specifically the structures of the R groups, to determine now what type of interaction we're going to see. All right, so if you look up the structures of leucine and valine, you will see that these contain non-polar R groups or side chains. So that means then that they are going to interact with each other with your hydrophobic interactions. Right, again, like dissolves like drive this, right? So if you have two non-polar things, they're going to want to be right next to each other. We have two cysteines, right? Cysteines contain that SH, the thiol R group. So that means then these can participate in a disulfide bond. If we have an aspartic acid and a lysine, right? Your aspartic acid is an acidic R group. So this is going to have a negative charge. Your lysine is a basic R group. That's going to have a positive charge. So we have a negative and a positive then, right? Those together will have a salt bridge, or just simply an ionic interaction between them. And then the last one, we have serine and we have threonine, right? If you look up their structures, what you'll see is that both of these contain the very polar OH groups between them, which means then that these can hydrogen bond or hydrogen bond with each other. Right? The only way that you can determine this is you have to go in and you have to look up the structures of the R groups to see what types of components they actually are made up of. Okay, so that brings us then to the highest level of protein structure, which is called quaternary. So quaternary is abbreviated 4 with a degree sign, and this is the highest level of protein structure, and this is actually maybe the simplest out of all of them. In order for a protein to have quaternary structure, that simply means that there must be multiple subunits. So there must be more than one subunits that interact with each other to make a fully functioning or fully functional protein. Right? So, for example, probably the most common example of a protein that has quaternary structure is something known as hemoglobin. Right? Hemoglobin is a protein you find in your blood that is responsible for transporting oxygen. And this Hemoglobin is made up of 4 subunits, 4 polypeptide chains. So we have one chain here that is in, we'll highlight it in green. We have another one that is also very similar to it. And then we have 2 more. There's a third one over here and a third one over on the other side. And each of those are able to carry 1 oxygen molecule. So we have 4 of these subunits that come together to make your fully functioning protein. Then we would say that this protein is going to have quaternary structure. These 4 individual subunits are held close to each other by the same types of interactions that you would find in your tertiary structure, right? So you'll have hydrogen bonds, you'll have disulfide bridges, you may have hydrophobic interactions that keep these 4 subunits together. So the last thing that we're going to address here that pertains to the structure of proteins is a phenomenon known as protein denaturation. So when we look at the denaturation of proteins, what we're looking at is affecting the structure of a protein. Specifically, when you denature a protein, okay, what you're doing is you're disrupting the secondary, tertiary, and quaternary protein structure. So when you denature a protein, the secondary, tertiary, quaternary structures are disrupted. And if the structure goes away, or the structure is not what it's supposed to be, right, that means the function of that protein is disrupted as well. And that's extremely important because proteins have to be have to have the correct structure in order for them to carry out their specific function. So structure dictates function, and that's a sort of a staple in biochemistry and biology. So if you mess with the structure, you mess with the function of not just proteins, but other molecules as well. One thing to keep in mind when we look at denaturation of proteins, right, denaturation does not affect the primary structure. Okay, it doesn't affect the sequence of the amino acids. That's still intact. What we end up affecting when we denature a protein is the actual shape of the protein. The secondary structure, the alpha helix or the beta sheet, the tertiary structure, the 3-dimensional shape, or the quaternary structure if you have multiple subunits. So what are some things that can denature a particular protein? Well, there's several things. Some of these you actually take advantage of, right? So heat and different types of organic compounds can disrupt protein structure by affecting and by disrupting specifically the hydrogen bonds and also the hydrophobic interactions. All right, so for instance, when you cook an egg, right, why does it go from runny to being really hard? Well, you're affecting the structure. You're denaturing the protein by providing heat. Similarly, you know, when you go to get an injection at the doctor, for instance, you know, why do they swab your skin with alcohol? Right, well, that's to denature the proteins and the germs that you have on your skin, for instance. Acids and bases can also denature proteins by disrupting the hydrogen bonds between your polar R groups, and they also disrupt the ionic bonds, right? The positives and negatives that attract each other. Remember, when you add an acid, you're adding H plus to something, right? And that can affect that attraction between a positive ion and a negative ion that you have in your R groups. And you probably use acids and bases, for instance, to help cook things, right? If you add an acid like lemon juice or lime juice to your raw fish, for instance, right, you can actually start that cooking process right away. Different types of heavy metal ions, so things like mercury, for instance, so Hg, or lead, for example, these can interact with your disulfide bonds, and they can disrupt those disulfide bonds permanently. Right, this is a permanent disruption, which means it cannot be reversed, which is actually a big deal, right? So if you are exposed to, you know, mercury or high levels of mercury or lead, for instance, it's a huge problem because it affects the function or the structures of your proteins, which will then affect their functions. And then finally, just by mechanical agitation, by physical mixing or whipping of a protein, you can actually start the denaturing process, right? When you whip an egg, for instance, you're actually physically stretching those interactions. You're stressing those interactions, and you're going to disrupt those interactions that keep the tertiary structure intact until those interactions are not there anymore. Right, so there are several things that can affect the structure of a protein, which will lead to its denaturation. All right, so as I mentioned before, when you cook an egg, for instance, right, you have here your egg protein, which is in its three-dimensional shape initially. Right, when you start cooking it, right, that shape is gone, because you've disrupted all of those R group interactions. So the R group interactions are disrupted. But you haven't affected the primary structure, right? So let's say, this was your N terminus over here, and this was your C terminus. You still have your N and C terminus. You still have the amino acids linked together. It's just that they're not in the correct shape right now, right? The three-dimensional shape of the protein is obviously not what it is compared to its active form. Your book has a really good summary in table 16.6 of all the different denaturing agents that you can use to help denature a protein and how they actually do that.